Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. If you're watching these videos, it's for one of two reasons. You're either enrolled in my class or you came across it in the inner Google. If you're watching these videos, please hit like and subscribe. That helps me, that helps other students, that helps keep it flowing and keeps it available for everyone. And as I add material or change it, you will get updates. Now, these videos are intended for students who are enrolled in my Human Anatomy and Physiology class. They are intended for my students to learn the material the way that I want them to learn it, and I keep it real simple early on. We'll get more complex as time goes on. If you're in someone else's class, either here in Corpus, at Del Mar, or somewhere else and across the US or the world, and you find these helpful, great. They may help you understand the more complex topics. What you need to do is you need to learn the material the way that your instructors want you to learn it. Read your textbook, add in those details that I may not be covering or may cover differently, okay? But some people say I'm smart, and I don't think I'm that smart, so I have to simplify everything to understand it. But it works for me, and it's very often worked for many of my students. So these videos are very often simplified explanations of some of the more complex concepts in anatomy and physiology. Now, in the last two videos, we went over the organelles and the cell story, how the cell functions, how all the organelles function together to keep the cell alive. We need to delve into a little bit more detail into specific organelles to understand some more of the more complex physiological processes that we're going to be discussing throughout Part 1 and Part 2A and P. And I can't emphasize this enough, and let me just tell you now, learn this material. It's not just about making an A on the test, because I know y'all are going for competitive programs and you're worried about your grade. But you're going to have to rely on this material later on in Part 1 and Part 2 and all the way through your career, and I'm not exaggerating. I just had a whole batch of students, literally a couple of dozen, graduate from nursing programs, either here at Del Mar or at A&M Corpus Christi, and all of them emailed me. I'm honored and flattered that they thought of me to say how much they utilize all of these concepts in anatomy and physiology all the time. So don't learn the material for the test. Learn the material for your future, because you're going to come back to these concepts over and over and over and over and over again, okay? Now, what we're going to start talking about is the cell membrane in a little bit more detail. Now, if you're following along in my class, you're using the note set that I write, and we're going to be on page 15 of my note set. We're going to talk about the plasma membrane or the cell membrane. So, when we talk about the cell membrane, it actually goes by a number of names. It goes by cell membrane, which is the same as the plasma membrane, which is also the same as the plasma lemma. Lemma comes from the same word as laminate, which means a layer. So it's a layer of plasma, or it's the plasma membrane or the cell membrane. We call it the cell membrane because it's the membrane of the cell. It's called the plasma membrane because it's very fluid or plasma-like and dynamic. It's not stiff and rigid. Now, we talked about the functions of the cell membrane before. It is a bilayer of what we call phospholipids, lipids that have some phosphate integrated into them. And so that's an important distinction between other types of lipids. But nonetheless, what I want you to know is this. The cell membrane has four major functions. We've talked about this in the last couple of videos: isolation, protection, permeability, and sensitivity. So the cell membrane provides all of this for the cell. It isolates the inside of the cell from the outside world. It protects the intracellular environment from the extracellular environment. It does control permeability. Anything that passes into or out of the cell must pass through the cell membrane. And it provides sensitivity. It can sense or detect the presence of certain substances and other cells surrounding it. How and why is a really fascinating concept. We'll get to it hopefully one day. Now I'm going to erase this part because you already really have that. That's a repeat, okay? Now, it asks you to draw and describe a typical phospholipid found in a human cell membrane. So some of what I'm going to talk about is a little bit more complex than I'm going to tell you, but just follow along here. When we look at phospholipids, they usually have a ring-shaped head, and we say that the head is hydrophilic. Anytime you have phil, it means to love. Like a bibliophile loves books, an audiophile loves music, a hydrophile loves water. So the head of the lipid is somewhat water-loving, okay? It likes to interact with water molecules given the opportunity. And then it has these long tails that look like these little crooked chains, and the tails, there's two of them, that are said to be hydrophobic. Well, if you have a phobia of something, it's a very strong dislike, and you don't like to interact with it. If you're afraid of snakes and I put a snake in the room, you might jump as far away from that snake as you can. So the electrochemical properties of the atoms in the tails are such that they don't like to interact with water. If I put the tails close to water, they'll flip and let the head interact with the water and not the tails. Now, these aren't heads and tails like you would find on a cat or a dog or an animal. Really, when we look at this head, what it is it's a ring of carbon atoms, and they can vary usually in their shape. And these carbons can have 4 bonds. Sometimes they have double bonds amongst each other. If I don't put the other bonds on here, you assume the other ones are hydrogens, but some of these have a little phosphate group attached, so they're called phospholipids. So the head is actually a ring of carbon atoms, and then some of these carbons actually bind to other carbons in these long chains like this. And there's sometimes double bonds and some, real unique chemistry. But they're long chains of carbons sticking off. These are hydrocarbons, carbons with hydrogen. So there's a ring-shaped head that's made out of a bunch of carbons and hydrogens and oxygens and some phosphate. And we have these long carbon tails. We just call it the hydrophilic head and the hydrophobic tail for now, okay? We're not going to get into that organic chemistry. I would love to, but that's a whole other topic in cell biology that you can take if you move on to a graduate program or something. Now, the reason this is important is because the heads love to interact with water and the tails don't. So if I dumped a bunch of these little individual phospholipids into a bucket of water and I stirred it up to where they were all floating down in here like this, eventually the tails would wiggle away from the water molecules and they would pop up and they would float on top like this. This is why oil floats on top of water. And some of them might even stick up like this, so that they will get the tails away from the water molecule, okay? So anytime you take some cooking oil, do this. Take a glass of water at home, put a little bit of cooking oil in there, whisk it up and break it into little bubbles, and wait a little while, and you'll see it all come together and start floating on top of the water. And if you do any chemistry, there's the organic phase, organic compounds with carbon and hydrogen, and then there's the aqueous phase, or the water, agua, aqueous. So anyway, so the heads and the tails, the heads like to interact with water, the tails do not. Now the reason this becomes important is because of this. If I take a group of these hydrocarbons, right, of these phospholipids, and I start to stick them very, very closely together, and I do this in a big giant circle, so that wherever I look on the outer edge here are all these heads with the tails sticking in like this. And then I took a second layer, so I have a bilayer of these phospholipids. And they're so tightly held up against each other that they're almost sealing everything out. And I went all the way around like this. And I did this in 3 dimensions. So that everywhere I look from the outside, I'm just looking down on the little heads of these phospholipids, tightly packed. But underneath them would be their tails. I could actually have water molecules all over the outside, and I could have water molecules all on the inside. But in this little layer in here between the tails, there's no water. Well, now we've somewhat formed the bubble or the membrane of a cell. We've just sliced it open and we're looking inside. Now I have an intracellular environment and an extracellular environment. So this bilayer of lipids is important for sealing the inside of the cell with fluid, the cytosol, and the extracellular fluid from each other. Now the cell membrane is far more complex than a bilayer of phospholipids. One of the things that we can see interacting with the cell membrane is some of these lipids have these little chains of sugars attached to them. And we call those glycolipids because they have glycogen or forms of glucose and other sugars attached to them. So occasionally one of these will have a little chain of sugars sticking off. And we'll look all over the cell membrane and we'll see all these sugars all over the surface of the cell membrane sticking off these little lipids. So now it's a little bit more complex. It's a phospholipid bilayer with some attached sugars on the external surface. Another molecule that can interact with the cell membrane is proteins. And when we assemble the amino acids, again, there's only 20 amino acids that all proteins are made from. But some of those amino acids have side chains. They have a chemical makeup that is hydrophilic. Some are hydrophobic. They don't like to interact with water. And when the proteins are originally synthesized, something that's kind of important to note, and you'll hear me talk about this a lot in the future, but suffice it to say, just pay attention for a second. Proteins are like long chains of amino acids, similar to beads on a necklace, all strung together with chemical bonds. It turns out that some of these amino acids can have little side groups, carbons and hydrogens and oxygens, sometimes some sulfur, that like to interact with each other in a chemical bond. And because these proteins are floating around inside of a fluid, if I bring them close enough, they can sometimes form a chemical bond and pop some water off. Well, that might bring two other amino acids close to each other. And these amino acids now, since they're close to each other, might form a bond, and eventually my protein might fold up into a three-dimensional structure. It's called protein folding, and some of those chemical bonds happen almost instantaneously if given the opportunity. To me, it's almost like, you know, those round windshield shades that once you start to twist them, they just collapse on themselves? It's kind of what happens here based on the chemical interactions of these amino acids. And some proteins can fold up in really weird shapes. And if I have a whole bunch of hydrophobic proteins in this region, they don't want to interact with water. And so sometimes these proteins can get embedded in the cell membrane, where part of the protein is sticking out here, part of the protein runs through, and part of the protein is inside the cell. And we can have proteins embedded in the cell membrane like this. And in some instances, those proteins can form little channels that allow substances into and out of the cell. They're called protein channels. Now, these kinds of proteins that are embedded in the cell membrane are called integral membrane proteins. Integral membrane proteins are integrated into the membrane. They're embedded in the cell membrane. Now, there are other proteins that, when they're synthesized, might loosely attach to the inside of the cell membrane based on their amino acid sequence. And a little bit of them might get embedded, but for the most part, the protein hangs out just on the periphery of the inside of the cell. These proteins that are loosely attached to the inside of the cell very often are called peripheral proteins. You're familiar with the term peripheral vision, your vision off to the sides. So these peripheral proteins are loosely attached to the inside of the cell membrane, where the integral membrane proteins are embedded or integrated into the lipid bilayer. Now our cell membrane is even more complex, okay? Our cell membrane is a phospholipid bilayer with associated sugars or carbohydrates, some proteins embedded in it called integral membrane proteins, and some loosely attached proteins called peripheral proteins. There's another molecule, at least one more that I want to discuss, that is important for cell membrane structure, and it's a molecule called cholesterol. Cholesterol is important for cell membranes, and it's an important part of our diet. You got to have a little bit of cholesterol in your diet and a little bit of fat. I mean, these lipids come from fats. So some people try to cut all the fat out of their diet. It's important to have a little bit of fat and cholesterol. It's a mix of things, not too much, but some. Anyway, cholesterol, since it's hydrophobic, very often can get embedded down in between the lipid bilayers inside of here. And one of the functions of the cholesterol, as it's embedded in there, I don't know if you can see this orange very well if I write too much, so I'm going to write it this way. But the cholesterol allows the membrane to remain somewhat fluid and flexible and not too rigid. So now, our cell membrane is a rather complex mixture of phospholipid bilayer with associated sugars and cholesterol and integral membrane proteins and peripheral proteins. And this is still simplified. It's more complex than this, but you can see how it's not just a simple thing. It's very much what we would call a mosaic. In art, a mosaic is when you take a bunch of different things and stick them together to form a picture. And this is a bunch of different stuff. The cell membrane is often referred to as the fluid mosaic model for this reason. The cell membrane is very fluid and dynamic. It's not fixed. And people don't see this at first, but when you study enough cell biology, you realize this. And here's what I mean. Let's say I take a basketball. And on the basketball, you know all the different sections with the little stripes. One part of the basketball has the nozzle here. It might say Spalding or whatever here, or Wilson or whatever brand. And it might say, you know, NCAA approved, and it might have the numbers for the weight and everything. If I look at this basketball, and I come back in a couple of 100 years, and that basketball's still there, that nozzle is going to be in the exact same spot in relation to all of this. The word Spalding isn't just moving around and sliding around. It's not fluid. It is static. The cell membrane is not that way. The cell membrane is very fluid. It's very dynamic. So, if I were to say, look down and see these two lipid heads next to each other, and there's a way to label these molecules. I've seen this in some of my research. If I could take these two lipids right here and label them with different dyes, one of them say with a red dye, and let's say the other one I use a blue dye. And they're in a specific location next to all these other lipid heads. And I got one that has a sugar attached to it there. If I came back in a couple of minutes and tried to look at this under a microscope, these little lipids heads to me are almost like looking down on a crowd at a very crowded concert. If two people are standing next to each other, one person might say, hey, I'm going to go to the restroom real quick, and the other person say, I'm going to go grab a beer, I'll get one for you. And so they might start to move in between all the people in the crowd, and if you were looking down on it, you would see the two guys in different colored hats moving in different directions. And that's what happens, is these little lipids can float around so that after a while, the one that has the red tag on it might be way over here. The one that has the blue tag on it might be over here. They're no longer in that fixed location. And the one that has the sugar on it might be moving. And the proteins that are floating in the cell membrane might be moving around sometimes. Not always. Some of these proteins are anchored to the cytoskeleton, but some of the lipids are moving around. It's very, very dynamic and always changing. Because of that, we have this definition of what we call the fluid mosaic model, which is in the note set on the same page. I think we're on page 15. It asks you, what is the fluid mosaic model? Well, this is what the fluid mosaic model is. It's this. It is the best working definition or description of the cell membrane. So the fluid mosaic model is the best working definition or description of the cell membrane. And it shows that the cell membrane is this complex mixture, and is a complex and dynamic mixture of phospholipids and associated sugars and cholesterol and proteins that are constantly moving. So it is a dynamic or complex mixture of phospholipids, carbohydrates, cholesterol, and proteins that is always changing and evolving. That's the fluid mosaic model. It's a mosaic because it's a mixture of things. It's fluid because everything's moving around or constantly changing. Okay? Now, it asks you to describe the different types of proteins that are associated with the cell membrane, and we've done that. Integral membrane proteins and peripheral proteins. And that's all we need to know for now. If you're following along in my note set for class, then we're going to move on to page 16. We'll finish page 16, and then I'll move on to another video. Okay? So now, we're going to talk about different types of transport inside the cell. And understanding the cell membrane is important for that, because some things can cross the cell membrane through these ion channels or these protein channels. They act like little tubes that can open and close and let stuff flow through. Kind of like the air nozzle on a little floaty or on a beach ball. And some substances can pass right through the cell membrane if they're made out of lipids. So we're going to talk about cellular transport. And we're going to talk about it in several videos because it's a long, complex topic. Anyway, I think I'm going to wrap this video up so it stays short, and then we're going to move on to page 16 in the next video. Thanks for watching. I hope you had as much fun as I did.